Hi, everyone. So I think we're going to get started in a moment here. I'm just giving everyone a second to return from break. So I think I'll get going. This session will discuss the impacts of receiving genetic information about psychiatric diseases on patients and on their sense of responsibility. Woo-Kyoung will be leading us off with a presentation of her work. Professor An is a professor of psychology at Yale University. Her main area of research is in higher level reasoning processes. Professor An's work addresses fundamental topics related to thinking, such as how people learn and represent concepts and causal relations, and how causal explanations shape our thinking processes. She also studies applied issues. For example, how expert clinicians' causal explanations for mental disorders affect their diagnoses, and how learning about one's genetic predisposition affects people's expectations for their symptoms. After Professor An's presentation, Larisa Heiphetz will lead a discussion. Professor Heiphetz is an assistant professor of psychology at Columbia University, where she leads the Social and Moral Cognition Lab. Her main research engages social and cognitive development, religious and moral cognition, and intergroup attitudes. So before I hand it off to Professor An, just a quick reminder, you can ask questions by typing them into the Q&A. You can ask those anonymously or with attribution, or you can directly raise your hand and I'll allow you to unmute and speak directly to our panelists. So Professor An, will you take it away?
Oops. All right, is this good?
Yes, great.
Thank you for having me today. And let me just first start out with the kind of responsibilities that I'm going to talk about in mental health management. One way to think about responsibilities in mental health management is to think of your body and mind as if it is something that one takes care of. So when a person is mentally healthy, then the person is responsible to stay healthy and avoid all sorts of things that are bad for you. When a person shows symptoms of mental disorders, however, the person should seek help by finding a clinician. And the person should also stick with the treatment plans and believe that the treatment is going to work. That is, she should be optimistic. So in the research I'm going to present today, I've been studying how learning genetic risks for mental disorders can affect all these behaviors. We now have much better understanding of genetic basis of psychiatric disorders, such as major depression, alcoholism, obesity, you just name it. But currently, personalized DNA tests, such as direct-to-consumer testing, including like the ones that are offered by 23andMe, do not yet offer tests for these kind of psychiatric disorders. But in the near future, they may start providing information for genetic risks for mental disorders for their customers as well. When that happens, there are two types of genetic feedback that people can receive. They can test positive for a certain mental disorder. So for instance, they may be told that you have elevated genetic risks for major depression. Or they may test negative. They may be told that you don't have a genetic risks for depression. And we can think of the impacts of receiving positive test results and negative test results separately. So let's first start out with possible impact of testing positive on these tests. When a person learns that she has genetic risks for, say, depression, one may think that it's not their fault to be depressed then. So in some sense, that's a good thing because it would reduce stigma about themselves as well as other people who have a depression and so on. But at the same time, people seem to have this misconception that genes are rather fixed. And this can make them believe that there's not much one can do about their problems. So to test this hypothesis, we experimentally manipulated the kind of genetic feedback that our participants received. So let me take some time to explain the methods that we used because all of the studies that I'm going to talk about today use the same method to manipulate genetic feedback. we manufactured our own saliva testing kits in our lab. Each kit contained mouthwash, and we mixed some sugar into it. Participants were first asked to rinse their mouth with this before performing the test. The kit also contained a test strip, and participants were told to place this strip under their tongue for several seconds. But this test strip is actually just a glucose test strip. But participants were told that it is a saliva test for detecting genetic risks for certain conditions. When participants put the test strip under their tongue, the color changed because of the glucose that was in the mouthwash. Participants were randomly assigned to different experimental conditions. Some were told that it means that the target gene was present, and others were told that it means the target gene was absent. The study was, of course, approved by IRB. Participants received extensive debriefing at the end of the study to make sure that they understood that the genetic feedback they received was made up by us. Participants could withdraw anytime during the study without losing compensation. But the compensation rate was the completion rate was nearly perfect. There were no reports of adverse effects in any of the studies I'm presenting today. This experimental manipulation of genetic feedback is necessary in order to rule out any confounds of actual genetic differences. This logic might be obvious to those who does a lot of experimental work, but just to make sure we're on the same page, I'm gonna illustrate this using this animation. So let's say a blue circle here stands for a person who is actually genetically predisposed to major depression, and a white circle stands for a person who is not. And let's say we could actually perform real, true genetic testing and could sort these people into those who test positive versus negative. And then we examine whether or not there's any difference between these two groups in terms of how pessimistic they are about their prognosis. And if we actually find out that those who test positive are more pessimistic about prognosis, we have no way of knowing whether this difference was because of the genetic feedback they received or because they're different in terms of actual genetic predisposition. Instead, what we need to do is to randomly assign various people into different conditions so that there are no actual genetic differences. And then some are told that they tested positive and others were told that they tested negative. And if we find out that those who received positive feedback were more pessimistic about their prognosis, well, then we can conclude that this effect is not because of actual differences in genetic basis, but because of the sheer genetic feedback that they received. So in the first study I'm presenting, Matt Leibowitz and I examined the effects of learning about one's genetic risk for major depression on prognostic expectations. At the beginning of the study, participants first took Beck's Depression Inventory, or BDI, which measures levels of symptoms associated with depression. In BDI, participants had to rate their agreement with a variety of statements such as, I feel sad all the time, I sleep most of the day, I can't concentrate as well as usual, and so on. And we only looked at the data from those who scored 13 or higher on this scale because this is a cutoff used to screen those who are mild to severe levels of depression. After taking the BDI test, all participants performed the saliva test that I described earlier. And in the study, this test was described to the participants as a saliva test for assessing genetic risks for major depression. Some participants were told that the results indicated that they do not have genetic risks for major depression, while others were told that that means that they have genetic risks for major depression. Then we used what we called a modified negative mood regulation scale, which basically measures the confidence in dealing with depression. So for instance, they answered to what extent they believe that when they're depressed, they will be able to find a way to cheer themselves up, or they would end up feeling really, really depressed. Compared to the gene absent condition, participants in the gene present condition were significantly less confident about dealing with depressive symptoms. Again, these participants were simply randomly assigned to one of these two conditions, but simply learning about the genetic test result caused this difference in confidence. In this study, there was actually a third condition where we tried to intervene on this misconception. In this condition, participants were told that they had genetic risks for major depression, but after receiving this feedback, they also watched an intervention video that we developed. And this intervention video was designed to dispel the belief that genes are immutable and to teach that gene expression is not deterministic. So here's an excerpt from the video.
Research has clearly shown that many factors affect whether or not a person becomes depressed. Some genes appear to affect a person's chances of having depression, but the truth is that genetics alone can never make someone depressed. Even among people who have an immediate family member with depression, a large majority do not become depressed themselves. This is true even though family members have a lot of genetic similarity. In fact, even if a person has a genetically identical twin with depression, most of the time that person will not become depressed.
Okay, so I think that was enough. And to remind you, learning that they have genetic risks for depression reduce their confidence in dealing with depressive symptoms. But as shown in this third bar on the chart, learning that genes are malleable and not deterministic restored this confidence. They were actually even more confident than even the gene absent condition. The first study that I just presented examined how genetic feedback can affect people's perspectives about future, what they can do in the future. In the second study, we examined how the feedback affects their memory for the past. Specifically, we tested whether testing positive on genetic risks for major depression can retroactively affect their memory for how much depression they had in the past. In this study, participants first started out with a saliva test that was disguised as a test for genetic susceptibility for major depression. And then, just like in the previous study, there were 3 different conditions depending on the genetic feedback they received and also whether or not they received the intervention video. The difference is that participants took the BDI test after they received the genetic feedback. In fact, this BDI test, which measures their depressive symptoms in the past 2 weeks, was our main dependent measures. And because participants were randomly assigned to one of these 3 conditions, there's absolutely no reason to believe that their levels of depression would be systematically different from each other. However, compared to the gene absent condition, participants who were told that they were genetically predisposed to major depression reported that they had experienced a higher level of depression in the past 2 weeks. And this time, the intervention video was not effective. The BDI scores was slightly lower given the intervention video, but it was not significantly different from the gene present condition, and it was actually significantly higher than the gene absent condition. So far, I presented 2 possible impacts of positive genetic effect. People may have more pessimistic views about their recovery because they believe that genes are fixed and there's not much they can do about this. And second, people may develop distorted memory about their past because they may believe that they are essentially a depressed person. And to frame this in terms of responsibilities, this means that people feel that they're not as responsible for their own depression. Now, switching gears, I'm going to talk about our more recent studies on impacts of receiving negative genetic feedback. So when people receive negative test results, for instance, when they're told that they don't have a genetic risk for obesity, it can lead to false reassurance. That is, they may believe that they're highly unlikely to have the disorder, and they don't have to worry much about developing that disorder, even though that is not necessarily the case. There have been similar phenomena demonstrated in the past in social psychology, known as licensing effects. So in one study, participants took sugar pills, but some of them are told that these pills were actually multivitamin pills. Then these participants were more likely to engage in unhealthy behavior, such as casual sex or heavy drinking, because they presumably felt licensed to do these things. In a rather old study conducted when anti-lock brakes were not that prevalent, they found that the drivers of cars that have anti-lock brakes tend to drive faster than those without anti-lock brakes. So similarly, we predicted that when they test negative in genetic testing for certain disorders, they may also feel somewhat invincible. In this study that I collaborated with Matt Leibowitz again, participants were randomly assigned to one of three conditions. In the first two conditions, they carried out a saliva test that was disguised as assessing genetic risks for obesity. And then participants received either positive or negative feedback. There was a third condition, and this time participants did not start out with the saliva test, so they did not receive any genetic feedback. This was just pure baseline condition. Then all participants were asked to rate significance of healthy diet and regular exercise in maintaining their weight. For instance, they were asked to what extent with the kind and amount of foods that you eat or the amount of physical exercise that you get influence your body weight. This chart shows the mean ratings on the significance of exercise and diet. The yellow bar shows the ratings by those who were told that they tested negative. These participants judged that exercise and healthy diet are significantly less important than those who received the positive feedback or those who did not receive any feedback. In this study, we also had semi-behavioral measures. We asked the participants to pretend that they have a day-long meeting on the following day, and they are to pre-order a meal. They're presented with 6 lunch options in the menu, and some of these foods were healthy, such as salad with grilled chicken, while others were unhealthy, such as meatloaf with mashed potato. And we looked at what percentage of the participants choose unhealthy food. The participants who were told that they were not genetically predisposed to obesity were almost 3 times more likely to select unhealthy food compared to the no test condition. And they are almost like twice more likely to choose unhealthy food compared to the gene present condition. What this study showed is that when lay people believe that they don't have a genetic risk for a certain disorder, they also believe that they're unlikely to have develop this disorder. And therefore, they don't need to do things to prevent this disorder. And this is irrational because in the case of obesity, negative feedback does not mean that doing exercise or eating healthy food is not important for maintaining healthy weight. We call this genetic invincibility effect. In the next study I will present, and that's the final study, we looked at another way that this genetic invincibility effect can be instantiated. This time, we tested the situations in which people already have symptoms of a certain disorder. And because of genetic invincibility effect, they may trivialize these symptoms and believe that it is not urgent to seek help, or it may not affect their functioning as much. In this study, I collaborated with Matt Leibowitz and Annalise Perricone. We examined the effect of genetic feedback on alcohol use disorder, or known as alcoholism. AUD is characterized by uncontrollable alcohol consumption despite clinically significant impairment or distress, and is among the most prevalent and undertreated psychiatric disorders in the world. Our study examined whether people who test negative for a genetic risk for alcohol use disorder might then treat the symptoms of an AUD as less serious and less urgently in need of treatment. At the beginning of the study, all participants were asked to take alcohol use disorder identification test or audit. And using the conventional cutoff for audit, we classified some participants as non-problem drinkers and others as problem drinkers. The main dependent measures of this study were AUD invincibility scores. Participants were told I see something on the screen. I don't know what it is, but I will just continue. Participants were told to suppose that they're spending a lot of time drinking alcoholic beverages, and they end up drinking more than they intended. And under this scenario, they were asked to answer a number of questions, such as, how serious of a problem would this be? Or how confident are you that you could stop these behaviors on your own without professional help or treatment? Or how urgent would it be for you to seek professional treatment for this? And under the same scenario, they were also asked to rate how much they would like to make changes in their drinking, or they should be doing things to cut down or stop drinking. There were 10 items like this, and because they showed high reliability, we averaged them to come up with the AUD invincibility scores. Participants first responded to these 10 AUD invincibility items. Then they carried out the saliva test that we used in the study that I just presented earlier. But this time, the saliva test was described as detecting their genetic susceptibility to alcoholism. And this time, everybody in the study was told that they tested negative. That is, they were told that the color from the saliva test indicated that they do not have genetic risks for alcoholism. And after that, they are presented with the AUD invincibility items again, to see whether participants became more invincible after receiving this negative genetic feedback. Here are the results from the pretest ones shown in blue bars and post-test ones shown in gray bars. Both non-problem drinkers and problem drinkers became significantly more invincible after being told that they do not have genetic risks for alcoholism. That is, even if they were drinking a lot and they're drinking more than they intended, they thought that these symptoms are less urgent, less significant, now that they believe that they're not genetically predisposed. This is a serious problem because it means that testing negative on alcoholism gene can cause people to drink more as they feel more invincible. In other words, testing negative can ironically become a risk factor for alcoholism. In this study, we also tested whether there's any way to lessen the genetic invincibility effect. We found that there are materials that are already in use by a direct-to-consumer genetic testing company. Specifically, when their customer tests negative about certain conditions like breast cancer, they provide debriefings to avoid false reassurance. This debriefing had been FDA approved, but it is not clear whether they had actually empirically tested whether the debriefing works. So we adopted these materials and used them as our educational materials for our participants. In general, the materials explain that the negative test result did not rule out all possible risk of developing AUD, and that the test did not assess for all genetic variants associated with risk of developing AUD, and that non-genetic, such as environmental factors or lifestyle, can increase risk of AUD. Half of the participants in this study received these educational materials broken down into two parts, just like in the actual company who uses this debriefing. They learned about other possible causes of alcoholism before they started the saliva test. And after they were told that they tested negative, they were further explained that there can be other environmental factors. And testing negative does not mean that there's no risk for alcoholism. For the non-problem drinkers, the educational materials prevented them from becoming invincible after they received the genetic feedback. This interaction effect was significant, and there was absolutely no difference between the pre-test and the post-test invincibility scores in the education condition. However, for the problem drinkers, the education materials failed to prevent the genetic invincibility effect. As shown in the rightmost set of the bars, they were still significantly more invincible after they received the negative test result. To wrap up, I talked about what can go wrong when people receive positive genetic feedback and negative genetic feedback for mental disorders. When they learn that they're genetically predisposed to major depression, we found that they could be pessimistic about their recovery. And they can also believe that they're more depressed than they actually are, which can further make them more pessimistic about their prognosis. When people learn that they're not genetically predisposed to obesity or alcoholism, they felt more invincible. And the intervention methods that are currently used to counteract such feelings of invincibility turned out not to be as effective for problem drinkers. These results suggest that we need more and better public education about genes. Lay people should learn that genes are malleable, and genes always interact with the environment, and genes are not necessary to develop a certain mental disorder, and there can be other non-genetic factors. We're still struggling to come up with a better intervention method to handle the genetic invincibility effect. Perhaps one possible way is that when the companies are presenting the negative test results, instead of saying that they do not have risks for alcoholism, the feedback can actually highlight what kind of risks they actually have, so that it would be more positive framing of the risks that they have, so that they don't feel less invincible about the results. And I want to thank Matt Lebowitz and Annalise Perricott, and also the funding agency that made this project possible. Thank you.